Bismillahirrahmanirrahim. The next topic in our chapter number 3, that is enzyme, will be the factors affecting the rate of enzymatic action. Which factors are responsible to affect the activity of the enzymes or the reactions of the enzymes. When enzyme react with the substrate, some important factors affect the enzyme, and they are needed to be in specific range to accelerate the activity of the enzymes. If those factors sometime become high, sometime become low, that affect the enzyme activity, or sometime it denature the enzyme. Denature mean when the activity of the enzyme completely stop, the structure of the enzyme completely destroy, and there is no more chances to accelerate or catalyze a chemical reaction for enzymes. So they are known as denaturation or denaturing of the enzymes. In these factors, first of all, we will discuss the factor temperature. Temperature, and this graph show the effect of temperature and the activity of the enzyme. When we heat up, when we give temperature to anything, any chemical compound, it start giving energy to that chemical compound, and their molecules start vibration. With that chemical energy, that kinetic energy, the activity of the molecules become high. So it is as the graph show that when we heat up, when we give temperature to the enzyme, the activity of the enzyme start with the fast rate, with the rapid rate. This increase show that when we give temperature or when we heat up the temperature, it increase the rate of reaction. This show the rate of reaction means when the rate of the reaction is increasing, it means that the enzyme work with the greater speed. So at specific temperature at which the enzyme activity is more, that is known as optimum temperature. At this temperature, the enzyme work with the greater speed. But if we further speed up the temperature, like this is the 40 temperature, which is of optimum for the enzyme, and when we further accelerate the temperature, like up to 50 or 60, the rate of the reaction is going down. Why? Because the enzyme at that temperature become denatured, and that temperature is known as maximum temperature. At maximum temperature, the structure of the enzyme disturbed. The bonds, especially the hydrogen bonds that keep the enzyme's tertiary and quaternary structure, it is disturbed with such high temperature, and the reaction rate show that it is going down. This optimum temperature for the normally for the human is 37 to 38 degrees centigrade, while for the other organism like bacteria that live in the hot spring, for that the optimum temperature is 70 degrees centigrade. So it must be changed from organism to organism, but the optimum temperature is that temperature at which the enzyme activity is high and the rate of the reaction is also very high. And if we talk about the minimum temperature, means that when sometime the temperature become very low, like it goes at the minus, so it will not denature the enzyme, it will just cause the to stop the activity of enzyme. And after when we give the temperature more, the enzyme will regain its shape, and then it will restart its activity. But high temperature denature the enzyme, and it will stop working permanently. The second factor that affects the rate of enzyme activity is the pH. proportion of hydrogen ion. And enzymes function most effectively over a particular pH range, and this range is very narrow for each enzyme. Sometimes enzyme work in acidic medium, sometimes work in alkaline medium. What the pH causes? pH causes changes in the ionic conditions. Sometimes it give more hydrogen, sometimes give more hydroxyl. And acidic means that it has more hydrogen which attach with the amino acids of the enzymes, and it can change the shape of the enzyme. As well as alkaline medium mean that it is giving more hydroxyl ion, and it can change the composition of the amino acid of enzymes. So each enzyme need a specific pH at which it work with the greater speed, and that pH is known as optimum pH, and that is different for different enzyme. Normally most of the enzyme work at the pH of 6 to 8, but there are exceptions. For example, protein digesting enzyme, both enzyme pepsin and trypsin, both work for the protein digestion, but pepsin work in the acidic medium with the pH of 2, and trypsin work at alkaline medium with the pH of 8. So that is a very narrow range in which each enzyme work at different pH. Another factor is the enzyme concentration. What will happen when an enzyme accelerating a chemical reaction and we increase the number or the amount of enzyme? The graph show very simple calculation that when the enzyme concentration increase, the rate of the reaction will be increased. Why? Because more enzymes are provided for the reactants. More enzymes are available for the substrate. When one enzyme accelerate the reaction of 10 substrate, its speed will be slow. But when more enzyme, we add more enzyme like 2, 3, 4, so it will pick up more rapidly the substrate, attach each substrate with the other enzyme, and then the rate of reaction will be increased, and we will get more product. So the enzyme concentration is the directly proportion to the rate of reaction, that when we increase the enzyme concentration, we will have more a rapid rate of chemical reaction. The velocity will be increased. Another last one factor that affect the enzyme activity or enzyme catalyzed reaction is the substrate concentration. When the rate of the enzyme keep constant and the rate of the substrate is increased, first at chemical reaction we will get more product. It means that the rate of the chemical reaction will be increased with the addition of the substrate, but at specific time the reaction will be no more, there will be no more change in the reaction. The reaction will be going with the same speed, having no increase or decrease. Why? Because when we occupy the substrate according to the enzyme, the rate of the reaction increase, because enzyme have active sites available. But when all active sites are occupied, there will be no effect of the more substrate addition. When we add more substrate and no active sites are available, there will be no change in chemical reaction, and this is shown by the graph. So all are these the factors which affect the enzyme activity or the rate of reaction which enzyme accelerate or catalyze.